Yeah, probably a little informed or go away a little more informed, or probably asking more questions and be able to read around the field. So in terms of the talks that we'll be giving you, so it's myself, we'll be introducing you to technological advancements and again bringing in the African perspective. Then tomorrow we'll have Tasniem Issa, this is Tasniem Salie. I also have a student called Tasniem Issa, but Tasniem Salie, who's a PhD student of mine. And interestingly, my field is medical genetics. I study the genetics and genomics of disease. But she's looking at what my colleagues in anthropology and archaeology, Alan Breyer, as well as John Parkington. I mean, she's looking at ancient human remains here in the Western Cape, interestingly, more than 2,000 years old. And she'll be talking about her own work, but also contextualizing what ancient genomes actually mean. So that's tomorrow. And the next day you will be hearing from Shareefa Dalvie, who's a lecturer in the Department of Human Genetics, as well as the Department of Psychiatry, talking about complex disorders. How are we approaching the genetics and genomics of complex disorders? And that will be sort of contrasted with what I'm going to talk to you about today, which is the genetic basis of Mendelian disorders, disorders that you see running in families reasonably clearly from one generation to the other. That's some of what I will be talking to you about today. Sorry, how's this sound for you? Is it okay? Is it better? Feedback? Okay, let's just drop this a little further and see. You may hear my tummy rumbling. But okay. Is that too soft now? Okay. I'm going to look at the two of you. Okay. All right. So that's what we're going to be talking about. So a sort of 30,000 meter view of today's talk is going to be about where we're located. Well, we're located at the University of Cape Town. Maybe get that out of the way. And I'll talk to you about, and much of what we'll be talking to you about is in fact not just theoretical. It's literally the work we do every single day. So which is going to be interesting rather than telling you about or reviewing literature with you. Tasniem will need to do that for some of the ancient DNA, but we quickly lapsed into the work we're doing ourselves. So I'll be talking to you about 3 different disorders that we've been working on and tell you what's come out of there. And then I want to skip into talking to you about how have we been engaged with, for example, the rest of Africa, growing genetics and genomics in Africa, and how has that come about. And maybe we'll have a second or two to touch on sustainability, and then we'll stop for discussion. Now, I used to also run something called the Darwin Seminar Series, and some of you in the audience were probably attendees at the medical school. And I'm sorry we've had to stop that simply because it took a bit of effort and it got a bit costly. But that's a story for another day. So I should probably start off much along the lines of what Tasniem is going to be talking to you about tomorrow. But this is just introducing the subject. This picture of a tree, which is rather colorful, is simply where I'm drawing your attention to the shape of the trunk of the tree and then the branches, just to illustrate to you the idea of human origins. So humanity originated here in Africa, and there's absolutely no question about it. Okay, so humanity originated here. And the important thing for me as a geneticist, and even when I lecture to first-year MD-CHB or medical students, I say to them that the value of studying the African genome is the following, and it's fortunate we have this kind of illustration to use. It might be obvious to you, you're all geneticists, because if I have to ask you, taking my looks into account, my physical features, where in the world do you think I've originated?
India.
Okay, so there's a geneticist in the audience. Okay, you didn't need to do any sophisticated genetics. That's my ethnic origin. All right, so we, and many of you in the audience, I couldn't tell where you originated.
India.
Okay, well, at least some part of you will have come from India. But I can't tell all of you, apart from probably saying the majority of you probably have your origin somewhere in Europe. Okay, I mean, I wouldn't be far wrong. My colleague here would obviously be from, or have origin somewhere in Africa, right? But the reality is all of us originated here in Africa. And I'm going to extend that example very simply. The value of studying African genomics is that when our ancestors left the African continent between 50 and 100,000 years ago, I mean, some of you in the audience will tell me it was 66,500 years ago, whatever. But whenever they left, some of us branched north and went to the left. Some of us headed northeast and went far east, our ancestors, etc., etc. And for those of us who remained here, I mean, I'm speaking about the indigenous African population. For those of us who left the continent, we only took with us a small subset of genetic variation that existed on the African continent. Okay? Does that make sense? So when you look at our genomes today, your genome, those of you, European origins and Asian individuals of Asian origin, our genomes are very much like the level of genetic variation is, for example, what you see on this institutional carpet. If you look at the floor, you will see it's reasonably bland. And I'm saying this with the greatest of respect, okay? It's reasonably bland. However, if you look at the genomes of indigenous populations in Africa, specific, not indigenous populations of other parts of the world, but of Africa, it's like a Persian rug. I mean, the richness of color, diversity, genomic variation. Okay? And that's where I'm going to stop with regards to explaining the value of studying the African genome. The African imperative for studying genomics is a different story. Okay, in other words, how are we going to use genomics to advance issues of agriculture, human health, etc. So when I'm explaining to folk what I actually, sorry, I don't have a pointer. I'm not sure if you have one. I can probably try and use the arrow for the pointer. Okay, can you see this? Yeah. Okay, so when we're looking at human diseases, I summarize it for colleagues. There are many reasons for many different bases for human diseases, but I look at cell biology. I mean, I'm a geneticist, but if I just simplify it, I look at what cells do. I'm not going to explain to this educated audience what cells are. If you look at this layer of epithelial cells up here, you can see the regularity. They look quite healthy, right? It's just an illustration. What I do is I look at either the incidence where cells are degenerating. So compared to that, can you see this cell line? You can see they're breaking up, disintegrating, right? Over here. As well as, that's on the one hand. And degenerative disorders are, for example, neurodegenerative disorders. And the examples I will give you of degenerative disorders is, in fact, blindness. And it's specific forms of blindness. I'm going to emphasize not all kinds of blindness, but a specific retinal kind of blindness, okay, or a retinal degenerative disease. On the flip side, here on the proliferative model, what I'm talking about is where cells are meant to be stationary or quiescent, okay, they actually end up growing more than they should, okay, and that's an example of that would be cancer. All right, so I'll give you an example of the work we're doing on cancers and so on. So just to get to the bottom of where we all began. So an example, the picture on the top left-hand side there is a fertilization of the egg by the sperm, right? That's the beginning of us as individuals. Okay, the picture up here. Now you know as much as I do that that particular cell, the fertilized egg, doesn't grow haphazardly in this instance. It does go from one cell to two cells to 4 to 8 to 16 to 32, etc., right? But it doesn't grow haphazardly in that way to emerge 9 months later as a big ball. 50% of you wouldn't be here. In fact, all of us wouldn't be here, right? The human species would be dead. Because if we developed in mom's uterus as a ball, okay, you'd have to explode to release us. So it's just not sustainable. That doesn't happen. But in fact, what happens is exactly what's shown here. Diagrammatically, the fetus actually grows very systematically. In other words, instead of this ball of cells, this shape of the fetus is actually occurs because only certain cells will grow at a certain time and other cells will die off at a certain time. Certain cells will differentiate into, for example, retinal cells or cochlear cells or cells of the gut or cells of the brain compared to just growing as epithelial or any other cell. Okay, that should be reasonably clear. But underpinning all of this, there is obviously a program If they were just growing, even as this big mass here, that there would be a program too. But you can imagine that the program that's underlying all of this, or some kind of conductor that's standing in front of this mass of 23,000 genes, is actually saying to a part of this particular orchestra, okay, wind instruments or string instruments, up, down, etc. There's some kind of process or program ongoing that actually results in only certain cells growing and developing at a certain time, other cells being downplayed, and others differentiating from one cell type to another. Okay, which is why you end up with this rather lovely looking entity here after 9. In fact, this is probably a child of 6 months at least. Sorry, I wish I had a pointer, but okay, you understand what I'm talking about. I'm talking about the level of development and differentiation that brain cells are where they are. You've got heart muscle cells where they are, red blood cells where they're supposed to be. Okay, that's a fairly sophisticated process which we obviously reasonably take reasonably for granted. Underlying all of that, as I said, is this process, and there's a picture in the middle that many of you in this audience would know. That's Boehringer Mannheim's pathway, or biochemical pathways. You would have had it in your classrooms, or at least you would have seen it in your children's classrooms, or whatever. It's a biochemical pathways. In fact, those are biochemical components and pathways which are all mediated by genes. Okay? The entire biochemical process that works in every one of our cells is actually illustrated in this. If it's not as clear as you think it should be, it's because it had not always been clear. And this pathway that was drawn in the 1960s, in fact, has grown in the level of sophistication. We know a lot more about it now. In other words, we know a lot more about those pathways. These are what happens to proteins, how proteins are formed, how proteins are formed are actually dictated by genes. So a particular gene will code for a type of collagen that will end up in your skin, your teeth, and your bones. Other collagens will end up in your cartilage, for example, and so on. All right? I mean, there are 23,000 genes that are working at different times and in different quantities. So obviously, if there's a potential for something to go wrong, I mean, look how complicated it is. There's a potential for lots to go wrong. And guess what? It does go wrong. It goes wrong in several different ways, and wrong is a relative word. I'm saying there is a potential for change, and that's different from wrong. The potential for change. And the potential for change is mediated by genes. So here's an example of why we study genetics. So this is also an example of an illustration I put out to first-year medical students. And I asked them, doctor, give me a diagnosis of that first picture on the top. I've got it there already. It's a stalled cow. All right? First-year students love being called doctor, by the way. So my question to them is, tell me what is wrong with a cow. This is a summary illustration. All the answers are there already, but I don't have it in their slide. I tell them, what do you think the problem is? Well, if they're medical students, or students generally, as I was, it's very likely it doesn't have petrol. Okay? In other words, my pocket money hadn't arrived. So it could be that there's a problem in the fuel system. But even if it is a fuel system, it could be any one of thousands of components along the fuel line, which is illustrated here all the way to the carburetor. Or it could be something with the electronic system, or the electrical system with the older cars, right? The net, I know many of you will be able to say, well, if you did this, you could get an answer there, etc. But I'm talking about, as a first-year medical student, you know very little about cars. You barely know how to drive them. Okay, so we as geneticists actually want to know precisely what is wrong and in which system is there something the matter. Because if there's something wrong here, perhaps I can intervene a little higher up and get the car started. And I'll explain to you how in genetics we're able to get to the precise point. And here I'm going to start talking about a couple of the examples that I mentioned to you. There's nothing wrong with this sunflower. Okay, so here's my predecessor, Peter Beighton. Some of you in the audience may know him. He was the previous professor of genetics probably about 15 years ago. Peter's retired, and he's probably about 85 years old now. Peter was also given the Order of Mapungubwe by the President of South Africa in recognition of the fantastic work he'd done. And some of this work was describing completely new syndromes that he'd seen and picked up in southern Africa. I mean, he enjoyed going to Namibia and different places, describing these different syndromes. This syndrome is called Sclera Cirrhosis, and I'm telling you this for a specific reason. It affects only as far as I know, I can't guarantee this, he certainly described it as affecting largely individuals in the Afrikaner population. It's a recessive condition. In other words, you need 2 hits of that gene defect, and it produces this particular phenotype. At that stage, and when he described it, we actually didn't know which genes were affected. And I'm going to describe for you very briefly how we went about finding the gene and the implications for potential therapeutics for another group of disorders. So this is a bone thickening disorder, so you can think about where it might be as a therapeutic as a public health intervention, for example. So sclerosis is this bone thickening disorder of general bone density across the entire anatomy. And what's illustrated here on the left is what these individuals look like. Physically, they look quite similar to one another. They're generally, there's this feature called gigantism. They're very large people. And their bones are radiodense, as illustrated on the skull here on the left, top right-hand side. Now, if those were just the issues, it would not be too big a deal. However, it can be a fatal condition. It's a lethal condition. Simply because these individuals' skulls, in fact, get hugely thick. Okay? And they end up dying due to brainstem compression. Okay? It pushes the brainstem into the neck. To stop that happening, these individuals have to undergo a craniectomy. In other words, a big piece of the cranium has to be removed and some kind of protective material is placed on the heads. That just relieves that aspect of it. So if it's a bone thickening disorder, we wanted to know what is the biology. That's the big deal about genetics. We want to find the gene, firstly to be able to use in clinic, and potentially to predict who else in the next generation may be affected with the disease, okay? And I'll tell you how we're using that in cancer. But in this instance, quite importantly, to identify the gene so that you can then figure out what the protein product of the gene does. If you have an idea of what the gene product does, you may come up with some kind of intervention for those specific individuals, okay? Some kind of therapeutic. There is a flip side in that you may not be able to help those individuals, but understanding the biology of what leads to bone thickening may be able to get you to a place where you can use that mechanism in individuals who are suffering from bone thinning disorder, which is called what? Osteoporosis. So for several years, we worked on this particular group of disorders, and what's illustrated here in a sort of hodgepodge fashion, but I'll walk you through it. On your top right-hand side are a group of families, and the shaded-in individuals, those circles and squares, illustrate the affected individuals. And you can see it's a recessive disorder. Mom and dad generally don't have it, but they end up producing kids who have it. So mom and dad are carriers of the disorder, of the defective gene. So what we ended up doing was looking at all individuals we could get across, let's say, 20 pairs with their parents, simply trying to find out, and I'm not going into the technology with you about how we end up mapping things onto the genome. It's quite a drab technology. But what we end up doing is looking with a large number of markers across the entire genome, 23 pairs of chromosomes with very unique addresses. These markers exist with very unique addresses. And look at what part of the addresses precisely are the affected individuals all sharing. But the unaffected individuals are only carrying one copy of it in this instance. For dominant disease, you're just looking for what all affected individuals are carrying. All right? And in so doing, you know that they're sharing a certain chunk of the genome. Okay? It's not like each gene is randomly distributed, but chunks of the genome move from one generation to the next. So what we're able to say is that all affected individuals are sharing this chunk of chromosome 17. And the next bit of work, as you can see next to it, is to try and find out what is the complement of genes that are lying in that particular area. All right? And in that era, it probably took 5 years of work in that era. This today will take you an afternoon. Okay, the genetic workup in the lab with next generation technologies. So effectively, just what are the, let's say, 10 genes that are lying in this area, and then you sequence each gene, which was a huge deal literally. This was done about 15 years ago. What genes are lying in this area? Sequence those genes. In other words, what are the base pairs, the building blocks, one next to the other? And what is the base pair that is changed, or is there a deletion or an insertion that is not present in healthy individuals? Sorry about the mic. So in that way, we're able to figure out through segregation analysis and say, this is the mutation. Because the mutation will give you the DNA sequence, which will then correspond to the coded protein and give you an idea, is this protein actually just have one codon change, in other words, one amino acid that's inserted, or in fact, is there a truncation mutation that may be stopping halfway to the coding of the protein? Or is it aborted right up front? Okay, I mean, that you will work out reasonably quickly. And as I say, you could use it for predictive testing and in genetic counseling, but quite importantly now, okay, it is this particular gene. We ended up calling it sclerostin, So that was the name that was given to it, reasonably arbitrarily. And the next bit was obviously quite important, as illustrated in this particular aspect of this slide, is to figure out what does this gene do, or the coded protein, biologically. What exactly does it do in the process of bone formation or bone degradation? Because it's a dynamic process, as you know. I mean, the maintenance of bone structure is dynamic. There's osteoclast and osteoblast building and breaking down bone. So we were collaborating with a group of individuals from Canada who then took it, not out of my hands, out of Peter Byton's hands, and said that they would like to develop potentially a therapeutic. And I mean, there were no contracts, et cetera, signed with the University of Cape Town, because this is very close to market now. And as most of you in this audience would know, the bone thinning disorder, osteoporosis, is a huge issue internationally in all populations. So this is being developed potentially as a therapeutic with absolutely no comeback for the University of Cape Town, or potentially no commercial gain, which is hugely unfortunate. And that just speaks to the naivety, that we have functioning in science. The university has woken up dramatically since then, and there's a whole issue of IP issues and whatnot. To some extent, it's a pain that you can't just work with your science, but you could be constantly suspicious of the motives of collaborators and so on, which is unfortunate. But that's just one aspect of the work that we're doing that has got potentially a very, very positive outcome. Sorry, do we have any water? It'll take me a longer time to go to the bathroom and come back. Will you get me some? Thanks. Okay, so the next disorder I want to talk to you about is cancer. You wouldn't say from this slide that I was going to talk to you about cancer, right? So this is a disorder that's called Inheritable Colorectal Cancer, okay, which is Lynch Syndrome in this instance. There are several types of heritable bowel disease or colorectal cancers. The one is familial adenomatous polyposis, and that's a dominant disease where, in fact, the astute doctor will actually trace this family as a dominant family, and would know that if you start looking into the colon of individuals in the early teens, you will know which individuals are likely to develop the cancer because their colons will be carpeted with polyps. All right, so they are premonitory lesions. The one that I'm interested in, or have been interested in for a long time, is this thing called Lynch Syndrome, where in fact there are no premonitory lesions. So the reason why I talked about genetic gems in the Macular and dust, that was just to be dramatic, but in fact the reality is we were first notified of this disorder by the mine doctor in a place called Clancy Mine. So Clancy is a mine that's far north on the Atlantic coast. It's probably less than 100 kilometers south of the Namibian border. And it's a De Beers mine, and we were called by the mine doctor, indicating that he was concerned that there were more individuals on his working lot who had this particular colorectal cancer occurring in individuals who were below the age of 40 and in many instances below the age of 30. So he was actually concerned. He certainly knew that two of them were siblings brothers of one another. But we could hear in that conversation that the mine authorities were actually concerned that these individuals may actually be developing the colorectal cancer due to some kind of occupational exposure. We read that into the call. And they were also worried because they were only seeing it in the males. In males, and I'll explain that was a misperception. So there's some background as to the disorder and why we're interested in it. Colorectal cancers, as you know, are a major burden of disease internationally. So when I got out there, this must have been in between 25 and 30 years ago, I came across this dusty folder in Peter Byton's archives. And I asked him, what was this interesting thing? And it had just gotten the paper from the South African Medical Journal that described this cancer as being affecting mostly males, and only males, actually. The first SAMJ article was only males in this community. But the reality was, when I got there with colleagues in genetics, as well as surgery, at that stage it was Dr. Mike Madden who was in surgery, and us in human genetics, got up to Clancy. In fact, what we'd realized was this wasn't Clancy. So Clancy, the mine on the coast, is about 50 kilometers away from this village called Tramachas. It's a Nama word that means something like water by a rock or something like that. You wouldn't know that, right? So I'm showing you this because we got to the mine doctor and actually interviewed some of the affected individuals and took a family history and then asked the question, well, where are the women? And they said, well, the women are not here. They're in our village 50 kilometers away. So the men are bussed in the morning. But in the bad old days of apartheid, the males were brought to the mine to work, and like security like you cannot believe. Firstly, trying to just get onto the mine, and getting into the actual digging area is just unbelievably secure, as it should be. But just these males were allowed into the property to do all kinds, even the gardeners outside of the actual mine. Women, the odd woman was brought in to do some kind of housework, etc. But interestingly, we didn't pick up any cancers. But when we went to the village, in fact, it turned out that the disorder was equipresent. In other words, in as many females as we were seeing it in males on the mine. So there was nothing about environmental or occupational exposure that we could speak of. So our work was done largely in these kinds of environments. And this kind of vehicle we had to use eventually, because in fact the disorder was far more widespread than just the mine or the village, Gamachas. In fact, is present in vast tracts of the Northern Cape. And I'll illustrate it in a moment. So if you just focus on, so many of these slides are composites because I wanted to get through as few slides as possible. If you look at this map, this is the southwestern tip of South Africa, you can see red dots. If you can see much here, you'll see red dots that are clusters. That simply illustrates the individuals and their families that we had picked up on these trekkings through the Cape. And Namibia, Comachas and Clancy are right up there. It's about 600 kilometers as the crow flies from here. So what I wanted to illustrate up front, before we did any molecular genetic work, we were managing that kind of lineage, that's a family tree, simply based on Mendelian risk. So an affected individual is obviously at 100% risk. Their siblings, as well as their kids, are at 50% risk. So if I'm affected, I've got this sort of look about my genome, a healthy copy of the gene and a defective copy of the gene, or a mutated copy of the gene, right? Which leads to me being predisposed. So I have a 50-50, or my kids have a 50-50 chance of inheriting either this or this. Do you understand that? Okay, so that's just taking some genetics for granted. So all we could do at that stage was simply take all of the individuals that were at 50% risk, not yet affected, and in some instances also their kids. The reason we started to take in their kids, although they were not affected, is in one instance we came across a child who was 16 years old and developed cancer and died of it. okay, colorectal cancer specifically. So that got us quickly changing our entrance criteria for the surveillance, and we started to look at much younger individuals, even if their parents were not yet affected. That had the net effect of filling up our colonoscopy clinics like you cannot believe. That's just a tiny pedigree, bit of the pedigree that we have up there. This pedigree would go around this room several times, just illustrating the lineage in the Northern Cape. So that was the reason. So we started off just going up for a one-day trip to Cleansheim Iron specifically. But after doing all of these tracings of the families, etc., into the wilderness kind of thing, we ended up doing about 4 or 5 day trips and probably stopping at 3 different hospitals. In other words, with our colonoscopy kits, etc., etc. And at this stage, we had Paul Goldberg, who was the head of the GIT, surgical GIT, who was part of this kind of surveillance program. Can you still hear me okay? Okay, good. So I then demanded that I take off time from the laboratory and from the Department of Genetics and took a year's sabbatical, because there was no ways I was going to get down to the deep, heavy-duty genomics research. And I went off to the University of Chicago Medical Center for just over a year. And the objective of that exercise was to produce this kind of figure of family tree. And I ended up, basically I wanted to find the gene, which was a formidable task in that era, to find the specific gene out of these 23,000 genes, and find the specific mutation that causes the disease. Because what I wanted to do specifically, or we as a team wanted to do, but I was the sort of sharp point of the pencil at that stage, was to be able to bring back a very simple test that of all the individuals who were at 25 and 50% risk, obviously I wanted to test the individuals who were 100% for validation, but then importantly to apply it to those individuals who were at 25 and 50% risk. And the big deal was to actually provide genetic counseling and release those who were mutation negative from follow-up. I actually never need to see you again. That was a very difficult concept to get across, because over many years we were insisting, if you're 25 and 50%, please come in for your colonoscopy. And they ended up being so compliant, it was amazing. So when I came back with this fancy test, and I had to tell the same individuals, I actually can do a test. At that stage we were just offering it on blood. You know, there's DNA isolated from blood. Now we'll do it on any bit of human tissue, like mucosal swab from inside your mouth. Said to them, okay, you're negative, you don't need to come back. And for about 2 years thereafter, these folks were sitting diligently outside because their relatives were mutation positive, were coming in. And they knew that they had to come in for the colonoscopy, because we said this to them previously. And they were confident that they were going to get the disease. They were literally fatalistic about this. I mean, they've been so used to seeing this run in their families and literally decimate their families. So now we were able to do exactly this. So this is a real live picture of what we are able to do. Affected individuals shaded. Red means you are affected and carrying the mutation. The individuals in yellow means you were at risk, either 50% or 25%. You are not carrying the mutation. Don't bother about, I mean, I'm being brute by saying don't come in. We literally counsel them very gently and got the message across, but obviously tried and didn't. And the greens are mutation. No, I'm sorry. The greens are mutation negative. The yellow is, it doesn't really matter, but for precision, green is you are mutation negative, don't bother. Yellow is you are mutation positive, you're on your way to becoming red. Okay? If you don't come in for colonoscopies, you're likely to be red. So what net effect has this had? Because I'm talking to you about applications. So these are survival curves. The one is survival curve, the fraction of 1, okay? And these are the ages in years. You can see mutation-positive individuals die off much more rapidly than their mutation-negative individuals, right? You can understand this. What I'm showing you here are both mutation-positives, but the top curve simply illustrates individuals who are mutation-positive and compliant with the recommended program of coming in for colonoscopies. Those individuals in red are non-compliant. Now, I'm saying non-compliant very broadly. In some instances, we may have never gotten to them, but in most instances, we did get to them and provide them with information. You are mutation positive. It's important for you to come in for colonoscopies, etc. And for some reason or the other, they may not have come in. You can see they die off as much, although they are mutation positive and tested, they would die off as quickly as their relatives who were not counseled previously. So that was literally where we were with that. And that's a program up to now. This is going fantastically. I mean, the program has increased remarkably. What we also know, for example, because we're following up these families extensively, that women in these families at a higher risk, the mutation-positive women, for endometrial cancer as well as for breast cancer. Okay, many of you would know that women with BRCA mutations in the BRCA1-2 genes, are at a higher risk for cancers. That's true, but this is another gene which has that same property. Okay, but not as high as the BRCA genes. Just the women here have a higher incidence. So in our surveillance strips now, in fact, what we have is the pink drive truck and staff. You've heard of the pink drive, is the surveillance program that is provided to women in rural areas. This is provided from an NPO called Pink Drive. I thought it was Think Pink, but I think that is something else. This is the pink drive that literally accompanies us on our colorectal surveillance trips to the Northern Cape. This work has been very, very well recognized. So my own input had won the Vice Chancellor's Alan Pfeiffer Prize for application in the community and making that kind of a difference. But quite importantly, we went on as part of an international collaboration, and this was with my friend, this is Sir John Byrne from the University of Newcastle in the UK. That's him and I, I'm not there because I'm taking the picture, visiting the village of Kamagas about 2 years ago. This was at the end of a clinical trial we had done around the world, including our communities here in the Western and Northern Cape, on very simple aspirin and starch. Okay, so mutation-positive individuals were on a double-blind placebo-controlled trial of aspirin and complex starch. And the net result was that there was a delay in onset. This was published in the New England Journal of Medicine as well as Lancet Oncology. So these are mutation-positive individuals. There was a net delay of close on to 10 years for those individuals who were on the aspirin arm of the study. So that was quite remarkable. As I said to you, these are mostly rural populations, and many individuals, perhaps even wanting to come in for colonoscopies, may just not have the means to. For the folk who work for the mine, we had an arrangement where they actually fetched bust people in. But it is also very inconvenient, because the night before you would have had a laxative or a cleaning agent that would literally ensure that your colon was as clean as the palm of my hand by the time you're in for a colonoscopy. And that's very inconvenient. In the middle of winter, for example, getting to the toilet, which is really an outside toilet about probably 1 kilometer down the garden or something like that. So it's really inconvenient. And they even have to stop the bus to go to the toilet on the way to come into the hospital. So many reasons may have mitigated for us getting that red line on that second curve I showed you. In other words, individuals not being compliant. So we've taken all of that for granted and thought this could be a very positive intervention. And in most parts of the world where the trial was conducted, this is already implemented. We're waiting for the Dosage Study to be completed, which is not being done here at the moment. It's being done in at least 4 other countries in the world where there is reasonably quick access to patients, etc. So these individuals were being given 600 milligrams of aspirin, which is a reasonably high dose, but in fact they had tolerated it remarkably well. I mean, we were looking out for complications, and they handled it remarkably well. So the next step that's being done and probably close to completion is the Dosage Study. And once that is complete, we'll be very happy to implement locally. How are we doing at the moment? Still attentive? Can I just have a glass of water? Okay, I needed somebody to keep time for me, please. Are you keeping time?
Yes.
How much of time do I have?
20 minutes.
20 minutes to go. Okay. let's try and rush this. Yes? In fact, that's less than 20 minutes then, right? Okay. Choose one. Okay, so I want to talk to you about an inherited form of blindness, and this specifically retinal degenerative diseases. Now, I bet many of you in the audience are familiar with, you may not experience this yourselves, but you know somebody with age-related macular degeneration. So this is a group of disorders that's related to age-related macular degeneration mechanistically, but this kind of retinal degeneration, or the inherited form of retinal degeneration, generally affects a younger population. I'm talking about a younger population overall, and let's say by younger, I mean less than 60 years of age. So mostly this is what I'm talking about. But I'm saying to you that we already know that there are a number of hereditary components that do impact on age-related macular degeneration also, right? So we've been looking at the genetic basis of inherited retinal degenerative disorders for many years, and all this diagram illustrates for you is for the different kinds of diagnoses that are made by an ophthalmologist, for example, macular degeneration, and a number of the others, Leber's congenital amaurosis, retinitis pigmentosa. There are several different genes we know already that lead to exactly the same phenotype, or what ophthalmologists would see through an ophthalmoscope. Okay, it's not just one gene. There are many different genes, each of which would be a mutation in each of which would be causative of the disease. All right, so it's genetically heterogeneous, this group. There's a clinically heterogeneous group of disorders, but even each entity there, for example, autosomal dominant retinitis pigmentosa is genetically heterogeneous. In other words, there would be many different genes, mutations in which individually would lead to the disease. Okay? I'm not saying you need a collection of mutations in different genes. I'm saying a mutation in any one of these genes would lead to that kind of disorder, and it would run in families. So these maps simply illustrate, for example, on the left-hand side, a range of different phenotypic groups of disorders, right? So Usher syndrome, for example, is in this pink. Blue is a sporadic type that we don't have a genetic marker for. Autosomal dominant retinitis pigmentosa is green. Macular degeneration is yellow, etc., etc. Stargardt's is this yellowish pink, and just keep your eye on that. This map is actually saying, after all of these years of research on this cohort, the research cohort, we have found this many genes. These are individual genes. So I mentioned Stargardt's disease here. Well, here's the gene for it. It's this pinkish part of the quadrant, or pinkish part of this pie chart, which says it has a mutation in a gene called ABCA4. So we know exactly what the gene is. In fact, the majority of individuals with Stargardt's, which is a type of macular degeneration, have a mutation in that. And all of the others colors represent, we found the genes or mutations in genes that lead to that particular group of disorders. So that's great. And the reason why it's important for us to have started off this research, first let me tell you that about 25 years ago, families who were suffering with this group of disorders, in fact, set up this organization called, I actually forget what it was called then. It was something like Fighting Blindness. But it is now called Retina South Africa, in line with their partner organizations around the world, like Retina International, Retina USA, Retina Canada. Okay? So it was a brother and sister team who started this organization. I won't mention the names, but they came over to see Professor Byton and I to say, look, when individuals with retinal degenerative diseases go to an ophthalmologist, literally all they're told is, yes, you are suffering from a visual defect, and I'll give you your measures, of your field of vision, etc., and all of the ophthalmological measures. The only thing that they could advise you was to go home and learn Braille, okay, and get visual aids to assist you in the meantime, and get your white stick, etc., which was logical and it made sense, but it came across very callously to the sufferers. And so they came to us and said, well, we've just been to a conference in Dublin, And what we've heard is that a gene for retinitis pigmentosa has been discovered. Not discovered, it has been mapped. Remember, I said previously, with those group of, we said we pinned it to one part of the genome. We still got to find out what the genes are in there. So a form had been mapped to the X chromosome in that era. And they came to us and said, look, we're convinced that there may be two other genes. The one will be dominant and the one will be recessive. Would you please undertake some research? We had no interest, in fact, very vaguely knew about retinal degenerative disorders. But we just saw the enthusiasm of these individuals, the lay public, so to speak. And we said, okay, well, you're presenting a strong enough case. And they undertook to recruit people from their organization and so on, and be able to help us work on the disorder. And what they had realized at that stage was, if they hadn't invested, if they don't, as the sufferers, invest in doing research in this group of disorders, in fact, 25, 30 years later, they would be exactly where they were then. They also knew it was the long haul. There was no quick cure for it. But what they were convinced about is the genetics would expose the biology. Okay? And with the biology in hand, we'd probably get to the next step, which was some kind of genetic interventions. Genetic-based interventions, not necessarily genetic interventions. So this slide, I think, works quite nicely. Simply what I've got here is, after all of these years of research, what we know clearly is that I showed you those overlapping Venn diagram kind of thing of different genes. What we know now, or what we expected, is that if I take you back to the analogy of the car, the stalled car, if your car stalls as a result of no petrol, you fitting on new spark plugs is not going to help at all. Okay, or changing the ignition system, or even you, if you've got a carburetor problem, installing a new fuel tank, although it's along the fuel line, is not going to help. And that's exactly what genetics is showing us, that you've literally got to be very specific with regards to which gene or gene product or biochemical pathway you're going to target. And this is a train station. I forget whether it's Japan or Paris, actually, I should know. And the trains look very similar. All I'm illustrating here is, if you've got a mutation in this particular gene called LRAT, it's just one of the genes in the retinal disorders, it's literally a big mistake for you to jump onto this platform and try to get onto this train. This is going somewhere very different. So in the field of genetics and genomics, this is a very good analogy with regards to precision medicine. You will hear this if you haven't heard it now. If you hadn't heard it up to now, you will hear it both in the media and in the press, this idea of precision medicine, that you've got to treat precisely what the biological issue is. It's an aspiration, I have to admit, but obviously this lot, the retinal group, are way ahead of the game. It's been a really long haul, but hopefully they'll stand to gain a lot. So here, for example, so already on the market, not on the market, sorry, in trials. I'm going to emphasize in trials, and mostly phase 1 and phase 2 trials, are a range of genes for the various different disorders that you'll see written up front, are these gene-based interventions that are currently being experimented on. Okay, so the one that stands out is the one for Leber congenital amaurosis, which is a retinitis pigmentosa in the pediatric population. In this instance, children are either born or exhibited very early in life. In other words, they're going blind very early in life. The good thing about this particular type of retinal disease, well, the good thing, the bad thing, I suppose, even for the one in the pediatric population, we know there are up to 11 different genes involved. So you've got to know precisely which. And in this instance, the second one, the RPE65, jumped out of the racing gates quicker than any of the others in terms of clinical trials, completely fortuitously, because Cornell University's vet school, veterinary sciences school, in fact had a dog population, an experimental dog population, in which, it was a model for retinitis pigmentosa, this dog population they had. Turned out that that dog population literally had a mutation in the RPE65 gene. Now, we're very similar biologically to other mammals. So in this instance, the experiments we do in mice, in dogs, etc., and the more evolved, larger animals we're talking about, the more similar they are to us in terms of our biology. So the fact that we had an animal model meant that the gene therapies could very quickly get through the animal models. That's the big deal as to why this is closer to market. And in fact, I know for now the RPE65 gene is the one that is approved by the FDA for clinical trials. So that's what's going on right now in several different centers. And we know that it has been reported to work in little children who are going blind, but specifically children with mutations in RPE65. This is an example of what happens. We use a viral vector, so a virus that we know would infect human beings, and hopefully it would be viruses that infect that specific target tissue. And what we do is we go in there and we remove the genes that are pathological or which confer pathogenicity to this particular virus, okay? And we just maintain the genes that are responsible for propagating them. So we remove a chunk of the genomes, and large enough for us to put a payload into. It's like literally a lorry, okay? So we clip a gene out of the human and clip it into where we have removed genes from the virus. I'm saying this because I'm also a microbiologist, so we literally do that. We can clip genes in and clip genes out. And those genes are then propagated, those viruses are propagated in a clinically appropriate environment. And obviously through various different quality control processes, ultimately purifying them and being able to, as I said, we tested these individuals, not we, had tested in the dog model, shown it to work, did phase 1 human trials, shown to be safe, and are now into phase 2 and phase 3 trials, which is hugely encouraging for, in fact, the entire field of retinal degenerative disorders. So I showed you the pie charts over there on the top right-hand corner with regards to, for example, one of the largest groups of disorders we have here in South Africa for retinal disorders is Stargardt disease. So this is a recessive form of macular degeneration. Both copies defective, so mom and dad will be fine, but mom and dad produced kids have a 25% chance of producing each pregnancy that has a child affected with Stargardt disorder. And they usually show signs of macular degeneration fairly early into childhood. So we've now been enrolled in a clinical trial that's going to be run between us and the University of Pretoria, simply because that particular institute, we don't necessarily want to be selfish because we're doing all of the genetics work here. The University of Pretoria and some of the private practitioners in ophthalmology have set up a really good program for clinical trials. So we actually want to make use of the best clinical trial facilities, which will fly past all of the hurdles fairly quickly. So that's set to get going by mid-year this year. So we're very, very excited about this. So all my point is, had we not done any of this work, people would not be looking at Africa at all, simply because this work has been done and published and has been showcased internationally that folk are attracted, like with the colorectal work I talked about, to come here and do the clinical trial with us. So our patients stand to benefit. Colleagues are very conscious of the time, and I was about to talk about the African dawn, which is literally, and all this part of the talk was about, I talked about some of the examples of the disorders and that we've been studying here, and all of that has been dependent on us having a medical genetics facility in our environment, or departments of medical genetics, and an interest like Peter Byton had. So we've, on that basis, developed a very strong molecular genetics research environment, a diagnostic laboratory, for example, that's internationally recognized, and obviously our clinics in pediatrics and adult health, that's really going really well. What I was going to illustrate to you, and I needn't show you the slides, and maybe I'll flick to them after just mentioning this very quickly. What has always been of interest to individuals such as myself is that literally, if you have to look at the map of Africa here, you'll see a red dot in Cape Town and a red dot where Cairo should be. And if you look at it's literally, that's where we have medical genetics facilities on the continent. So whenever we talk to our colleagues about some Cape to Cairo initiative, it's sort of laughable, but it sounds really exciting, and we're setting up education training programs. But if we, and I remember about 15 or 20 years ago attending a South African Society for Human Genetics meeting in a place called Clarence in the Free State. And I stood up and I said to my colleagues, I've joined the African Society for Human Genetics. This is a continental organizer, which nobody had heard of previously. There was a South African Society for Human Genetics. I think it would be a wonderful idea to join this society because I think we need to show the relevance of genetics and genomics for the continent or encourage the development. I didn't want to be, somebody came across as being patronizing about Africa. I didn't intend to be. And the questions I got from my colleagues in that room in Clarence were, but what has Africa got to give us? What has Africa got to contribute to us? We're okay. And that annoyed the life out of me. It was literally not what I had meant. I mean, what I meant was us reaching out to other parts of the continent.